Hello, my name is Mail Jindi, and I'm a research associate in the lab of Dr. Jeremy Teo at New York University in Abu Dhabi. I have a master's degree in immunology from Yale University, and I'm currently focused on immunobioengineering and the effects of microgravity on the immune system. Human space travel has become an increasingly popular area of interest, and it has long been known that there are many adverse effects of space travel on the human body. One of the most affected systems is the immune system, but there is still a lack of information on how and why this happens. Determining the causes for a weakened immune system during spaceflight is critical in supporting astronauts in future long-term space travel. T cells are one of the key players in the adaptive immune response and are critical in fighting off diseases. Any dysregulation in their functions can lead to increased rates of infection and chronic illnesses. These cells exist in circulation in the peripheral blood as well as in organs such as the spleen and lymph nodes. However, most studies focus on investigations involving traditional 2D cell culture or samples from the peripheral blood of humans. This provides an inaccurate representation of tissue-resident T cells due to the critical interactions between the cells and their microenvironment that are lost in 2D cultures. Our study utilizes 3D collagen matrices as well as 2D cell culture to better mimic the microenvironments of both tissue-resident and peripheral T cells. In addition, T cells were studied in their resting and activated phases. These cells were exposed to simulated microgravity conditions using a random positioning machine. In this study, we utilize our engineered biocompatible microvessels that allow for high-throughput studies of smaller sample sizes without the need to use larger flasks on the RPM as traditionally done. Our findings revealed that samples in 3D cell culture were less affected by simulated microgravity conditions at a transcriptome level compared to those cultured in 2D. In addition, simulated microgravity has less effect on activated cells than resting T cells. We also performed computational fluid dynamics that reveal that all the effects seen are due to simulated microgravity and not due to any fluid shear stresses on the cells. This leads us to believe that 3D cultured T cells, or tissue-resident T cells, are quote-unquote protected from the effect of simulated microgravity by their microenvironment. Also, activated T cells appear to be less plastic and more differentiated, therefore less able to change according to the environmental changes. We recently reported similar results seen in fibroblasts that are less subject to simulated microgravity effects when cultured in 3D collagen matrices. This study, along with our subsequent study on dendritic cells under the effects of simulated microgravity, are the first of their kind that study cells under simulated microgravity conditions in 3D cell culture as well as traditional 2D cultures. They are also the first studies to perform RNA sequencing on samples in these conditions. Further studies should focus on determining whether the effect of 3D cell culture maintaining the transcriptome profile of T cells is a temporal effect. Shorter or larger cell culture periods would allow for the determination of a specific time point when this protection comes into play and when, if ever, it wears off and cells are again affected by the microgravity conditions. More studies should focus on culturing cells in microenvironments that mimic their physiological state as opposed to simple 2D cell culture conditions.